Last one before break. It's kind of tough to be, but I like to also talk about details. So, Mr. Chairman, cut me after 15 minutes. Sure. Okay, I would like to basically continue with the study that was similar study that was already presented this morning. We also cooperate with Oak Ridge National Laboratory. We did some study on poplar wood together with a colleague Adrian Cernescu and team of AutoCube. And I would like to present today part of the study that was published just this year related to natural poplar variants and how it's connected chemistry of the cell wall to some other properties. So it will be basic outline, give you some backgrounds. We'll have a slide of objective, one methods, result, conclusion. So we will deal with, we deal with samples with the materials that is biological. It's a cell of the wood samples. It has some structure, known structure related to one cell that is bind to the other one. And basically, we wanted to know. It's good to know how the material is connected with the composition, with some other properties that it performs. So we know that it's a chemical composition of cellulose. Hemicellulose is lignin, some extractives and other components. Minor. These are detectable, but indirectly you can do some staining of samples. You can see where it is and what's the distribution. For example, there is a nice autofluorescence of a poplar cross-section on the picture, and highlighted region is full of high concentration of lignin, for example. So we know what's the position, what's the quality and quantity, but how they relate to other performance, for example, mechanical or some physical material properties, it's a question. The other slide is the middle one. This one, it's related to mechanical properties, elastic modules of elasticity within this cell wall or part of the cell wall, and but exactly what we see, we don't know what's the chemistry. So question is how those individual elements, composites, the composite element of biocomposite contributes to the mechanical performance or some other performance. So objective was to study those relationship between composition and of the cell wall and mechanical properties at the nanoscale. As a material, we use 3-year-old poplar species, Populus trichocarpa. They were typical with the content of the lignin. So we have high lignin sample, 27% of lignin was material, low lignin, it was only 22%, and there was also a mutant that has a natural mutation to single nucleoid polymorphism. That affects lignin content and the structure, of course. So it has also naturally lower lignin content. And then we took the samples, we prepared them for the atomic force microscopy analysis. We embedded them in epoxy medium, in spore. There was an idea that cell wall is impermeable to the epoxy medium. That has some larger structure. It doesn't get inside the cell wall. So, but it's needed in order to polish the surface. We use diamond night in order to eliminate surface roughness as much as possible. And we did some scanning through the cell wall. We use two regions all the time. The secondary cell wall, or this is the wall, secondary cell walls together like a compound, and also compound middle lamella, which consists of primary walls and middle lamella. Those are very especially good detectable, and the position or the boundary is very well detectable, so it lines up much the results from the distribution of chemical compositions, you will see on the picture later, and also mechanics change very rapidly at the boundary, so it was good to do. As observable method, we use nano-FTIR from Neaspec. Those are all data that are related to, much better about this. It was presented yesterday about the methods. For me, it's just a black box, and it works, I believe it, because we saw some good correlation. Outputs of this device is absorbance spectra at the space within a cell wall. And of course, also there is a mechanical amplitude and mechanical phase, and the phase is, we will see later, somehow related to, and it was also presented to viscoelasticity, to some mechanic performance of material. We run those scanning on 3 samples, this high lignin, low lignin, and mutant 3. We did some line scanning. I think it was 40 or up to 60 places within a cell wall. Then we process data, of course. Thanks to provided software, Neaplot, we run some baseline correction, and also using Omnic from Nicolette. There is a really powerful tool because it kind of allows us to do macros, and we could process data in a better way. Just write a macro, run this procedure, and go for the coffee. We did 2 corrections. One was correction between peaks. We did 2 corrections. One was for correlation between peaks and mechanical properties using this moving deconvolution process. And then we normalize data based on maximum peak between 3 samples within each sample. And then the other procedure was done for special quantitative comparison of chemistry, and colleagues from Oak Ridge laboratory, they did standard normal variate transformation, which means every spot was treated or normalized separately in order to get even data. So data before transformation and after shows the results within 1 sample. The other technique what we use, we own multi-mode atomic fluid microscopy, and they have from Bruker nanosurfaces, and they have a really good device or tool, big force quantitative nanomechanical tool, nanomechanical mapping tool. You know probably this approach curve. So we did this peak force is based on the approach and approach curve, which means you push the tip into the surface into the certain peak force setting point. In our case, it was 250 nanonewtons. And from the unloading curve, you are able to obtain elastic modulus, apparent elastic modulus based on the MT model. So outputs of this, and of course another feature is the adhesion force that is needed in order to remove the tip from the surface. So output is spatial modules of elasticity within a cell wall. You know what's the adhesion force, you know what's the deformation, how much go inside the tip, and also you know something about dissipation energy that tells you something about viscoelasticity or how the material behaves, how much energy is consumed during this approach process. So let's go to results. The main result is on this slide. Basically what we took the specific wave numbers, and we correlate them with the mechanical phase from nano-FTIR analysis. And the reason why we did it is that this mechanical phase is somehow related also to mechanics of the surface. It tells you something about viscoelasticity, plasticity was also mentioned. We saw that it is in inverse relation to stiffness of the material, which means those 3 peaks that are listed on the image, those are related to polymers, polysaccharides, cellulose, which is structural component, and hemicellulose within a wood. And those 3 are the main or the most correlated with the mechanical phase, which is reciprocal, and we can say that higher nano-FTIR peak, we got even higher elasticity or higher stiffness, let's say, of material. When we take a look at the spatial distribution within those 3 wood species, and we pick lignin functional groups, those are on our image right now, and we saw that there is a lower amount of, or lower peaks within a secondary cell wall, and very much higher peak was since within a compound middle lamella. We also saw that those high lignin tree, poplar, got also higher values of lignin peaks within a compound middle lamella, which was expected, of course. The others got a similar result, peak. When we take a look at the other polysaccharides functional groups, that are related mostly to cellulose and hemicellulose, and we saw that it was a reverse problem. So secondary cell wall, very much high content of those groups, and compound middle lamella was lower in both peaks. And when we took a look at 997, which is related to cellulose, we got similar result, very high values of secondary cell wall and smaller in compound middle lamella. But there was a feature that there is a cellulose in compound middle lamella, although sometimes there are some approaches that saying that there is no cellulose in middle lamella, which is not true, not the case. Okay, hemicellulose was the same. When we took data, when we analyzed data from peak force QNM, we also saw what is the distribution of modulus elasticity and adhesion within those 2 regions. And this is the adhesion line, which is higher adhesion at the secondary cell wall related to functional groups of polysaccharides, and lowering secondary compound middle lamella. And with the modulus of elasticity was the same, higher in the secondary cell wall because of the structural component like cellulose that gives stiffness of materials, and lower in secondary compound middle lamella. We wanted to also know what's the significant difference among the species, high lignin, low lignin, and mutan. And we saw that there is no significant difference. Basically, there was a difference only between those 2 regions, but within those 3 species, secondary cell wall perform the same way. And also compound middle lamella shows a little bit decrease of the elasticity due to increasing of lignin content, but those changes were not significant. We conclude this that maybe the variation or the differences between lignin content was not much good enough in order to perform for lowering the stiffness of the wood, or this part of the wood cell. So when we conclude this, quantity of polysaccharides of course predicts mechanical properties. We saw the coloration, huge one. I forget to mention that 46% of variation within peaks is contributed to variation in mechanical phase, which means that there are some other factors, of course, that there are some other factors that contribute to changes. Spatial position and quantity of polysaccharides and lignin could be identified in those 2 regions very easily. And there is also one conclusion that There is a higher, of course, modulus of elasticity, higher stiffness in secondary cell wall due to present and orientation of the crystalline cellulose. And we find out that there is no effect of lignin content on elastic modulus. Okay, almost, I passed 15 minutes. So acknowledge, would like to thank the agency that support this research. And I also would like to acknowledge the other authors of the paper that we published from Oak Ridge National Laboratory, from Haar University, and also Philippe Schaffer from AutoCube, and Cenk Geryo and Artur Rakuakas from Oak Ridge National Laboratory. Also thanks is given to people from Nicolet, Jan Vavra and Karel Šes, because they help us to analyze data, help us to understand how to process the nano-FTIR data. So thank you very much for attention, and if you have questions.